Okay, so today we're going to focus on a real-world analysis using bedtools, just to give you a sense of the types of things that we can do and some of the limitations of these types, some of the challenges, I guess, was these types of analyses. Okay, um, so that's what we're going to work on. So the question that we are going to focus on today is something very specific, and we're going to walk through some of the challenges doing this. So last time one of the things we downloaded was this Chrome file for human embryonic stem cells that, if you remember, the goal of Chrome is to take this RNA-seq and transcription factor binding site information, lots of different assays, and try and segment the genome into constituent pieces, parts of the genome in human embryonic stem cells that are poised for transcription, are enhancers, or are promoters, etc. And what we're going to focus on today specifically is enhancers in human embryonic stem cells predicted by this program called Chrome. And the thing that we're going to try and ask is, for all the enhancers that exist or predicted to exist in human embryonic stem cells, which of those are under the greatest constraint? And constraint is a bit of a loaded term. What I mean here is have the least genetic variation. So they're arguably under purifying selection. So if you have mutation or genetic variation in these enhancers, it perturbs their ability to regulate gene expression. That's the concept. So enhancers that sort of have a normal amount of genetic variation, we would say, are sort of neutral. Things that have a lot of genetic variation, we'd say, well, they're very tolerant or they're not under much purifying selection. But enhancers that have very little genetic variation, we might predict. We won't be able to prove that today, but we might be able to develop a metric where we could predict enhancers that may be under purifying selection. And that is what I'm calling constraint in this case. Right. So the first thing that we have to do is figure out where are our enhancers. Okay. So the materials that we need, first of all, we need a set of predicted enhancers. We could get this from ENCODE directly. We could also use this Chrome file that I use. This is more of a bigger question of like how you actually define what an enhancer is. But let's just trust these predictions from Chrome for today. The question we're asking, and that's just a bed file, right? That's just a bed file of intervals. From here to here on chromosome 22, Chrome predicts it's a promoter. Down here, it predicts active transcription. And maybe way down there, later, more distal in the chromosome, it predicts, hey, there's an active enhancer. But what that bed file doesn't have is genetic variation. We have to get that from somewhere else. So what is the format? Just thinking back to your last homework, what's the format that stores genetic variation? VCF file, right? For this exercise today, so that we can download files in a reasonable amount of time, we're going to focus on VCF files from the 1,000 Genomes project. Remember that despite the name, it's actually not 1,000 genomes, it's 2,504 genomes. So what we're going to actually be able to do is compare genetic variation observed among 2,504 individuals from all over the world, take those genetic variants and essentially intersect them with enhancers to figure out which enhancers have the most, the least, or the sort of middling amount of genetic variation. And that's this third step. We essentially need to count the number of genetic variants per enhancer. Does this make sense? Okay. All right, so we already downloaded this file the other day, so we don't have to do it again, but just for reproducibility, if you forgot it, you can download it again. But if you haven't logged into Malibu already, let's do that and just make sure that that file is there. If you don't have it in your directory, if you deleted it by accident, download it again. So today, just to make these commands run in a reasonable amount of time, especially because I'll probably make mistakes and have to run it a couple times, we're just going to focus on chromosome 22 as a pilot experiment. If we trust that the results look reasonably good on chromosome 22, we would maybe expand this pilot to the whole genome. And if you're super excited about this after class, maybe you could expand it to the whole genome. I suspect you won't have time for that given capstones. But just to remind ourselves, let's just take a look at this file. Oh yeah. Can you guys see in the back? Ish, a little bigger. Oh yeah, what do we do? Oh yeah. Better? Yes. Oh, because I didn't tell it how many lines, right? That's a nice error message. Okay, so this fourth column is that annotation. It's the prediction that Chrome-HMM made for each of these intervals. Okay. So how might we go about pulling out the subset of intervals that in this file that are enhancers, predicted to be enhancers? Any guesses? Yeah. Yep, we could grep. I'm going to show you two ways of doing this, okay? But the first would be to grep for enhancers. But what I would argue is if you've never seen this file before, one of the first things you want to do is what I would call profiling your data. Get a sense of what are the distinct values that are actually in that column so that you know what to grep for. And I think this is a mistake that a lot of us make all the time. You sort of are so excited about just like trying to get an answer. We often don't spend enough time actually plotting data, profiling the data to get a sense of if there's any weirdness in the data. And so this is, I would argue, a really critical first step. Does this data set actually make sense? All right, so let's see if I can get out of here. I'm just going to steal this command. We're going to look at that fourth column from this file. And actually, I'm going to do this a different way. I'm going to do this with awk so that we remember awk. So what's the analog in awk to cut-f4 before I type anything? Say again? $4, but what do we need to do with $4? Print it. Yeah. So let's forget awk. We're forget cut. We don't like that anymore. Let's just stick with a better program. And okay, so that prints the fourth column. There's a lot of them in there. How do we get to this? You've learned this trick already. It's on the slide, but this sort unique trick is a really, really powerful tool to just get the distinct values in a column. So the unique command assumes that the data coming in are grouped or sorted by each individual value, and then it's just looking for changes in the values, assuming that it's sorted. Okay, so if I sort, we know what this should look like. So it's unique is just going to scroll through that like we did with our eyes and just catching when it changes. Okay. Okay, and then if we do unique-c, we get a count for each of these. So it looks like the most common thing in here is a weak enhancer, if I'm counting right. Looks like it. And it looks like there's 4 different types of enhancers that are predicted. State 4 from this HMM is a strong enhancer. State 5 is also a strong enhancer. There's nothing in that name that tells us why one is strong or less strong. I'm going to guess that this is the strongest based upon the number, but there's nothing in here, unfortunately, that tells us. So we could, at this point, this is a decision in the analysis, right? Do we include both weak and strong enhancers? We probably, if we wanted to make a decision about that, this is the point where we'd probably read the paper and figure out what they actually mean by strong or weak. what are the decisions that went into that label, and how do those decisions connect to the biological question that we're trying to ask. Okay. But now at least we know what we want to grep for. Okay. I think I'm just going to guess. I haven't read this paper in a long time, but I think we should focus on strong enhancers because that's arguably going to be the more confident prediction. It also is a smaller data set for this class analysis, so it should go a little faster. So if we grep for strong enhancer, that should really include that category 4 and category 5, right? So let's see. I think I might have missed a step here. Oh, yeah, I wanted to subset the full file to just chromosome 22. I don't think I did that. So let me do that first. And just a reminder, this caret sign means that we're grepping for this CHR22 at the beginning of the line. Okay, so make sure you do that. So now we've got all the predictions from chromosome 22. Then I can grep for, I'm going to cheat, for strong enhancer. And I'm making a new file, which is called chrome22 enhancers.bed. And I would say that that's a lazy naming on my part. Let's fix this and let's call it strong enhancers so I know what I actually did. And let's make this font a little smaller. Okay. So let's make sure that that worked. Trust but verify. Looks like a mixture of only strong enhancers. How could I prove it? I could do a cut or an awk on that 4th column, look at the distinct values, double check, but I scrolled through it and I didn't see any changes, so I'm going to trust it. I just want to show you one other thing with awk and hide family photos. Let's see. I guess I can't. Sorry, guys. I just want to make this visible to everyone, but maybe next time I'll choose a different background. So we can grep for strong enhancers, but you can actually do the same thing. You can actually do slightly better grepping with awk. And that's called regular expressions or pattern matching. I'm just going to show you how to do that quickly. We can say awk. I always forget how to do this. I might have to Google it, but I'll try. I'm going to say $4. And now I'm going to introduce a new symbol. It's this tilde, squiggly sign. And that's a pattern match. And what you do is you put your pattern between 2 slashes. Okay? And let's see if this works. Let's do this from the full original file. So it's a real test. Yeah, so that works. What's slightly better about this? Well, twofold. One, we're saying exactly what column we want the match to exist on. With grep, we weren't able to do that. It's scanning the whole line. So awk, you can actually have finer grain control, I guess, over the matches that you get. The other thing that awk allows by putting the pattern that you're looking for between these slashes, you can do more complicated regular expressions. So we had a crash course 5-minute introduction to regular expressions with grep a month ago or a month and a half ago. I would recommend looking into awk and regular expressions if you're doing any sort of pattern matching with files, looking for motifs in files, looking for particular HPO terms if you're interested in that kind of stuff. This is a great way to just sort of slice and dice files and search in a slightly more sophisticated way, perhaps, than grep. Okay, so I'm going to create a new file. Okay. All right, so we have our enhancers now. We have our strong enhancers. And now, unfortunately, I'm going to have to on-the-fly edit all these commands, but it's okay. Right, so we have our enhancers. The other experimental data that we need, that we decided a few minutes ago, is genetic variation. What's great about this, we'll probably fill up our hard drive, fill up the hard drive on Malibu doing this, but let's go for it. We're going to download this file from 1,000 Genomes. One of the greatest things of the 1,000 Genomes project was transformational in many ways. It's absolutely publicly available data. Everything is available. It's one of the only human genomics data sets that's absolutely free to the world. And the other thing that came out of it, obviously, is the definition of all these different formats like SAM and BAM and VCF and all these tools that have empowered all sorts of other things. But for a class like this, it's fantastic. We can just go download all the genetic variation observed among more than 2,000 people from all over the world. When you think about that, that's pretty cool. So we can copy and paste this command. For me, we're all now going to fight for wireless for the next few minutes. I got there first, so mine should go a little faster maybe. Let's just give this a minute or two just to make sure everyone has been able to download it. But in reality, we're not actually doing this wireless here. It's all happening on Malibu, so it's all fine. Is it going similarly quickly for you guys? Okay. We're probably pushing the bandwidth of what Malibu's connected to. Malibu's right down the hall. All right, so let's wait another minute. Does anyone look like it's going to be an hour before it downloads? Is it sort of like a minute-ish? Okay, I think I'll proceed, but if I get beyond like 2 slides and it still hasn't downloaded for you, raise your hand, let me know, we'll pause. All right, so this is a big honking file name. It is a gzipped file. I just ran less on it and it worked. I think that's because my less command is actually alias to zless, which allows you to scroll through a gzipped file. I haven't checked that, but it's just a VCF file, like we saw before with Freebase. The only difference here is that there's no genotype information. It's just the sites. So we get the first 9 or 10 columns. So we get the chromosome position, which is relevant here because we're taking the variant positions and intersecting them with the positions, the intervals defined by our enhancers. And we get the reference allele, the alternate allele, and then some information about how frequent that variant was among the 2,504 individuals. But mercifully, for this course, we don't get all the genotypes, because if we had all the genotypes, this file would be massive and we never would be able to proceed. So they give you, 1,000 Genomes gives you 2 flavors of files. One that is just the positions where genetic variation exists, and then 2, the positions plus all the genotypes of all the individuals if you want to do a deeper analysis. Right. The other thing we want to get is not just the VCF file, but this tabx file. This is the index into this VCF file that allows us to use a program called tabx below to grab genetic variants from a particular chromosome, for instance, chromosome 22, or a particular chromosome and subinterval on that chromosome. I'll show you how to do both of those. So let's download this additional file. Whoops, I botched it. Okay, this is much smaller. Now that we have the VCF file and the tabx index thereof, the two together can be used by tabx to do to random access into different chunks of the data set. And the chunks are defined by chromosome or chromosome intervals. So let's just start with seeing what this looks like. One thing I want to note is I'm also asking for this -H. That says, hey, when you print out the variants on chromosome 22, which I've asked for down here, also print out the header from the VCF file so that the output is a valid VCF file itself. because a valid VCF file requires the header. If we didn't do that, when we go to intersect the results with the bed file using bedtools will say, hey, I have no idea what this file is because it doesn't have a header in it. If you put the header in the output, bedtools thinks it's good to go. All right, so if we do that, oh boy, we don't have TabX. That's a joy. All right, let's see if I can quickly install this for everyone. Okay, I gotta remember my password. Okay, we're going to compile it. We're going to take a quick break here while I fix this problem. Okay. Please hold. Where's TabX? Oh. Oh no, never mind. Okay, can everyone type TabX and make sure it works for you? Not Yabix. Does it work? Okay, cool. All right, moving on. Let's see what other gaffes I make here. All right, so I got to go back to this directory. Okay, so when I run TabX now in Chromosome 22, the sanity check we're trying to do here is make sure that TabX, we're going to trust that it works, but we always want to verify that it works. So let's look at the output, and if it's working the way we expect, the first record that comes out after the header should be for Chromosome 22, and sure enough, it is. Okay. One thing I just want to show you as well is that I can do this. I can say Chromosome 22 from, say, position 50 million to 55 million, and look, it gets variants that are roughly in that. Why is it doing this? I think I don't really know. It's just before 50 million. It must think, oh, this is a copy number variant. So the start is below 50 million, but the end of it comes into position 50 million. Okay. So you can use TabX. TabX is really, really nice for just pulling out subsets of data based upon a gene that you're interested in, if you know the coordinates or particular chromosomes, etc. All right, so now we want to save that to a tiny little VCF file called, I just called it 1000 genomes chromosome 22.vcf. So if you copy and paste that full command, it'll take a second. And now we've got this much smaller file, right? So it's probably something like 3 to 4% of the original file. So it's basically now we've got this enhancers file and we've got our genetic variation. So now we have to look at the coincidence of the genetic variation and those enhancers to get a sense of which enhancers have sort of a typical amount of genetic variation, which have a lot, which have a little. And we're really focused on which are the ones that have very few genetic variants. Right. So one thing we could do is count. For each interval in the enhancer file, we could use this -C option, which we learned last time, which is counting how many genetic variants from this 1,000 genomes VCF file intersect each enhancer. I've given away the punchline here. This is not going to work, and we're going to talk about why in a second. I think you probably already know. Huh. All right, I'm just going to have to type it out. Okay. So it doesn't like the fact that it says something about this file has inconsistent naming convention for this record. What that means is when it says naming, it's talking about chromosome naming. Probably after class I should update this error message to say inconsistent chromosome naming. So why is that? Well, look, the enhancers say CHR22, but the VCF file, anyone remember what the VCF file said for chromosome? Just 22. bedtools doesn't attempt to say, oh, I guess they mean that those are the same, because that can lead into all sorts of problems. If it starts to guess that 22 is the same as CHR22, you could imagine situations where that might not be exactly the same, so it'll give you wrong results. So it's assuming that the chromosome naming in all the files that you're trying to intersect or work with is consistent. If it's inconsistent, it's going to complain until you fix it. And really, you want to have consistency. So what do we do? How do we handle this? Any ideas? This is a common problem. You will face this problem. Yeah?
Can you use VCF tools reheader?
I don't know. You might be able to do that. If it will, I don't know if it will go in and update the chromosome labels. Maybe. That might be an option. I'm not familiar with that one. Yeah?
Can you just do awk on the enhancer file and replace chromosome 22 with just 22?
Could do that. Yep. Or we could do awk on the VCF file and update the 22 to CHR22. The easiest of those two options is probably just to update the bed file because it only has data lines in it. The VCF file has that header and data line, so the command to modify that is going to be more complex than modifying just data lines. Okay. So there's a couple ways to do that. I think you proposed awk, is that right? So how would that look?
Let's say awk print, or have the tab separate OFS.
Yep.
Tab, and then print 22, comma, and $2 dash.
Yep, $2, $sign.
I guess you do 3,
4, yeah. See what that looks like. Oh, but it needs a file. Huh? What am I doing wrong here? Okay, that worked. So that's one option. We can use awk to update the chromosome column to just be 22 and save this as something else. Like we're quickly getting into very hideous file names, which means we're doing real work. So we could do something like this, like fixed chrome. Okay. Another option that I think this next slide presents. So this is we've already realized this. The chromosome labels are different. The other option introduces another command called sed. Yeah.
So I've still been unable to use tabbix.
Huh.
It's giving me saying the index file either does not
Oh, did you download the tabx file?
Yeah.
Do a ls-ltr in your directory and look at the time, the date and time associated with each file. Ideally, the .tbi file has a later timestamp. Why don't you delete it, download it again, and see what you get. Is anyone else facing that problem? You are too? Huh. Okay, yeah, I'm not sure why that would be. The timestamps.
We can just have them tabs.
Yeah, the other thing you can do, if that persists, all you need to do is say tabx and then the .gz file, and it'll create an index for you. Just re-index it, okay? Good idea, Mike. Okay, so sed is a new command. We've not learned sed, right? It is new. Some of you may know it. Sed stands for stream editor. It's another, cryptic name. But what it allows you to do is essentially edit input data, and it corrects that data. It's kind of like find. I think of it as find replace. Obviously, awk can do the same thing. Sed can be sometimes simpler and faster to do these kind of find replace operations. So the way this works, in this case, what we're trying to do is rip off that CHR from the beginning of the line. So what this says is, hey, stream editor, I'm about to give you an expression, -e. The expression, just like awk, has to be surrounded by single quotes. Okay, and this is where I hope to convince you that this is like find replace. When you're in Word and you do find replace with misspelling some word all over the place, you have to tell it what it's looking for. That's between the first pair of slashes, and what to replace it with. That's between the second pair of slashes. Okay. In this case, I'm looking for the letter CHR that starts at the beginning of the line. So there's some consistency here. Caret is beginning of the line. And we want to replace it with nothing. So just essentially deleting the CHR. Okay? And then we end quote. Now, if CHR existed in multiple places in each line, and we wanted to replace all of them, we do something different. We would get rid of this caret sign because we're searching for any instance of CHR. And then at the end, what we'd say is global. We want to replace all instances of CHR on that line. If we don't have that global, it means just find and replace the first one that you find. Okay? And that's what we want to do in this case.
Sorry, I may have lost track, but do you need to change it to the strong enhancer?
Yes, gee, I do. You're right. Thank you. Question is how I edit this line now. Thanks for catching that. Pardon me? This one's right. If I fix it. Yeah. The limitations of editing slides in real time. Okay, we're good there. Bad here. Thanks for that. Okay, so now what we're going to do is not string. Wow, not 120. That's pretty cool. Okay. Right, so now we have this new file. The results should be the same. So how do we test whether or not the awk solution and the sed solution gave the same result? Any ideas? So of these two files, one I called fixed Chrome and one I called no Chrome, no CHR. How do we know that they're the same? Yeah. Oh, I thought you were answering.
Good.
Yeah.
I mean, there's strong indication because their files look the same.
Yeah. So a good hint is that the byte number is exactly the same. But I'd like to do even more than that. Yeah.
Diff.
Yeah, there's a program called diff, which will tell you if it reports nothing. So you give it 2 files. And if it reports nothing, they're the same. Okay, so there's no differences. Let's test that though, just to show you. Let me hack the first one. And it'll tell you there's a difference at the first line. Okay? All right, let me fix that. All right, so now we've got a chromosome 22 enhancer file which has the same chromosome labels as our VCF file. So we should be in business here. So we can, I gotta fix this one too. Not string, strong. All right, so now we can run it. Did your tabx index files fix? You're good to go? Okay. Okay, so looks like I blew something up here.
You put in strong enhance.
Say that again? Oh, thank you. Is that right? Oh, we'll find out, I guess. Okay, so now what we've got are, here's an enhancer with 25 variants in it. Here's another with 60. Here's another with 2. So of this like first snippet, which one looks like it might have the most constraint? Any ideas? Constraint we're defining as the least genetic variation. Yeah, so this last one only has 2. And it is a 200 base pair enhancer. And there's 2 variants. So the question is whether or not that's any different. Is that really different than this one, which is 8? So there's 8, but you know what? This enhancer it's 200 base pairs, so that actually is the same length. So that would be the density of genetic variation. So the number of variants per base pair is equivalent, is lower in this one than in this one. But one of the things we need to account for is the potential that some of these enhancers are longer than others. So the count itself is a bit naive. We need to convert that into some sort of density, right? Do you have a question?
Do you have to take into account if the enhancers are repeated throughout the genome? Oh, I guess we're only on chromosome 22, but like if that enhancer
The sequence itself? Yeah. Right. So that's a subtlety we won't address today. What I do know from this file is that the intervals do not overlap. So basically, chromosome or Chrome-HMM partitions the whole chromosome into distinct segments. There's no overlap. However, a really strong enhancer on chromosome 22, underneath the interval, it might have the exact same nucleotide sequence as an enhancer elsewhere on another chromosome. But I think what we're assuming here is that these are cis regulatory elements rather than trans. And for this experiment, I would want to ignore that, but that's definitely a follow-up experiment that you could do to see how redundant, how common that sequence actually is in the genome. Okay, so the problem is that some of these, I just talked about this, some of these are big, some of them are really, some of these are small, some of them are really big. So we have to account for the size of the region. So this is again where we can use awk. Well, let's convert that count into a density. So density in this case is nothing more than the number of variants per base pair. So all we do is take the count of variants divided by the length of the interval. Agree? Okay. So that is, let's make this better. So we're doing the way I've shown you. So what we're going to do is take that output for every one of those lines. That we now have 5 columns: the chrome start, end, enhancer label, and count of variants. We're going to print out that full line, and then we're going to take that new 5th column, which is the count, and we're going to divide that by the length, and we're going to have a new column that is density. Okay. Let's just, before we write it to a file, let's have a look, have a go at this. Thank you. Where does it go? There? Okay. Thank you. And then this guy as well. Lesson learned. Right? So now we can really directly compare these values. And I think what you see is that these numbers are more consistent from enhancer to enhancer now. The dynamic range of values is, the variance of these values is much lower because we've actually normalized these counts to a density value, which accounts for the variable size. This is a common thing that we have to do in genomics, or really any type of analysis like this. If you don't normalize the signal, then you get a lot of false positive predictions. Okay. Right, so we've got, let's run the full command here and save it to a what I'm calling a bed graph. That's simply because now we have some quantitative values associated with each of these intervals. It's strictly not a bed graph because bed graph only has one quantitative column, which would be the 4th column. But I'm contributing to the abuse of these formats. Let's make sure it looks right. Cool. Okay, so we have density. Now what do we do? What can we do? We could sort. Yep, so let's do that. Let's just take a look. What do we sort on? What column? 6. We want it to start at 6. We want it to end at 6. We want to sort how? Numerically. And we'll just take a look. Ooh. So this one's fairly biggish, and there's no variance. The density is 0. So maybe this is like the enhancer that makes our brains or something, you know? Maybe it's super fancy. Or maybe it's an artifact that we don't know. So what's at the end of the spectrum? So we got a dynamic range of 0 to 0.1. And just looking at this, given that, I'm going to guess that the mean is something like 0.05. Okay? But I'm not a savant. I can't look through this and sort of picture what the distribution looks like. So if you've not done this type of work before, this is the next step is, all right, we've normalized, we've sanity checked the data. Now we want to visualize what the distribution of these values look like, because what the distribution looks like governs to some degree what type of statistical analyses we can do. We might be able to do. So if values look roughly normally distributed, then there's all sorts of nice tests that are convenient, like z-scores, and you can do things like t-tests if things are roughly normal. If it doesn't look normal, all is not lost. It's just you have to consider the different types of statistics that you might need to do. Okay. So I think this will be our first foray into R. So this is what we just did. We looked at that. We sorted this by value. Now what we're going to do, I don't know if R is actually installed on here. R is not. So let's take a look and see if we can install R. If not, I can just do it on my laptop and we can follow at home. All right. This might not take that long, actually. Spend 30 more seconds on this and then give up. Oh, God. Cool. Cool. Didn't do anything. Pardon? Is it? I don't think we have modules on here, but I will try it. Nope. Okay. Well, we're gonna switch over to my laptop, and I will install R after class. Okay. No, it's strong, right? Not string. Thank you. Takes a village. All right, so I'm going to download this file to my laptop. One more time. All right, fine. All right, so I'm just going to steal this. Okay. R has a command called head as well. You just have to put things in parentheses, which I haven't done yet. There. Okay. How many of you have ever even opened R before? Oh, awesome. Okay, so most of this is not going to be review. All I'm doing, I have a file. It's this text file that we put on Malibu. I copied it to my computer because Malibu is bare bones and doesn't have R yet. I'm using this read table function to read this data file in as an object, a table, and I'm telling it that there's no header because we didn't put any header in the bed file. So we've just got these variable number 1, 2, 3, 4, 5, 6. Okay. What I'm going to do though is just build a histogram. So there's a nice function in R. I guess you can't see this in the back or to the right of me. We run the histogram command on this data frame that we just loaded. This syntax says do it for all the first number between the brackets is the row numbers. If you leave that blank, it's for all rows. And then we're using column 6, which is this density score that we've calculated. And I'm creating a histogram with 50 bins or breaks in it. And I like this, I'm obsessed with this Dodger blue color. So I do everything in Dodger blue, and then I add my labels, okay? So we get, wow, it's like thumbnails now. Check that out. The font is, if you can read that, you should join the Air Force. So this is the variant density. Looks like the mean, so here's our 0.1, here's our 0.0. It looks like I was wrong. The mean is more something like 0.3, and since the font is illegible, we'll just compute that directly. So we'll just say mean of that column. Okay, the mean is 0.03. We can count this, calculate the standard dev. Oops, not the standard deck. Oh, what is it? I always mess it up. It's an extra D.
It's just SDE.
Oh,
It's just
yeah.
SD.
SD, okay. So the standard deviation is 0.01. And, you know, this looks not really normal. It's somewhat symmetric about the mean. One of the limitations with this is a normal distribution has no constraints in the positive direction or the negative direction. We have an inherent boundary at 0.0. So this isn't really normal, but we can get a sense that these values down here are actually pretty extreme with respect to the broader distribution of variant density observed across all these enhancers. So this is where magic starts to happen a little bit. Like what do you do? How do you define what is interesting? And that's way beyond the scope of this class, but one thing that's commonly done is if it looks like this, you could just convert these density scores into percentiles. What density score is in the 99th percentile? What density score is in the 0th percentile? And then you can start thinking about, well, if I compare the properties of enhancers with variant density in the 1st percentile versus the 99th percentile, are there interesting differences between them? But that gets into other controls that you need to do. Make sure that those properties aren't the same as like enhancers in the 50th percentile, et cetera. Okay. Everyone with me so far? Okay. So this gets at another problem. You know, this looks kind of cool. I would probably, if I had the luxury of being a grad student again, which that would be awesome. It would be cool. I would be excited to take this plot and show it to people in the lab and talk about it in a lab meeting. But people would probably ask, well, like, what sanity checks did you do? Same thing. Are you sure that's right? Like, how do you know that there's not something weird about? So what about these regions with zero variance? So one, I think, truism for not only genomics work, but any scientific work, if you're looking at percentages or counts of things, anytime you see something where 100% of the data is there or 0% of the data is there, red flag. Something that might be real biological signal, but Occam's razor says it's probably, there's some artifact, there's something weird reason why you got 0 or 1. So look closely at those. So that would be one of the first things I would do, is maybe go to the UCSC genome browser, look at those regions that have 0 variant density and ask, is there anything weird about that? One thing that you could imagine is maybe that enhancer, maybe Chromosome somehow predicted an enhancer in a region that's like all ends in the reference genome. Maybe it messed up. I would want to rule that out. The person that wrote that program is a really good programmer, but everyone makes mistakes. All this software is written by people. Double check it. Are there any other artifacts or things that you guys can think of that you're concerned about? I can't think of any. I'm really just asking. Yeah.
So, coming from experience, we're going to look at the lengths of all of these things to make sure that they're reasonable, like the annotations are correct, so we don't have anything way too long.
I like it. Let's do it. Let's see if we can. So how would we do that in R? Let's do a histogram of the lengths. So instead of the histogram of the density, we can do a histogram of the lengths, which are column 3 minus 2. Oh, that's standard deviation. Blah. I messed something up, apparently. What? I can't even see what that is. Oh, yeah. Okay, I know what I did. Sorry. Doesn't work this way. So we got to take the third column, minus the second column for each row. Okay. Man, I'm not sure. I think it has to do with the projector. I don't know why I have no idea what those numbers are. Most of them are less than 600 base pairs. And then, yeah, there's a few that are greater than 1,000. But the dynamic range is something that looks like maybe 200. Well, we know there's 200 all the way out to 2,400. And then maybe something like 3,800. I honestly can't say much about whether or not that passes a sniff test. That again might be worth comparing other enhancers predicted by other methods, like annotated enhancers from ENCODE or from Vista as another project, that to see if this range is within sort of what you'd expect. But that's definitely a good thing to check. Okay. So that's one analysis. We're going to stop there for enhancers. What I want to cover now is a broader question, which is, what if we just wanted to look at constraint for arbitrary windows of the genome? So this was a very focused analysis on enhancers that are predicted by something else. But what about just literally taking the chromosome and splitting it up into equal-sized chunks and seeing if there's arbitrary regions that have less genetic variation than others? Okay, so that's actually, it turns out to be a very similar process. The only thing we don't know how to do right now is define chunks of a genome, chunks of a chromosome. So pieces 2 and 3 are the same. Piece 1, material 1 that we need, reagent 1, is this bed file of overlapping 10,000 base pair windows that are tiled across chromosome 22. So what we're going to do is basically, if this is the chromosome in black, what we're really trying to do is create chunks that are staggered. There's a whole class of statistics and thinking about whether or not you make them overlapping, non-overlapping windows, like just this top row of blue windows, or stagger them. For reasons that I won't go into today, except just to say that this gives us a little more signal to detect, makes it a little more sensitive so we can detect fine scale constraint changes. We are going to stagger these windows such that it skips. If the window size is 10,000 base pairs, the next window will start midway through, or 5,000 base pairs into the first window. And then we'll move forward 5,000, 5,000, 5,000, and each window is 10,000 base pairs long. Okay. To do this, though, we need to know how long the chromosome is, right? Because we could start, if we say we're saying we need 10,000 base pair windows and there's a 5,000 base pair step along the way, we know where to start. We start at 0, but we don't know where to end. That genome file that I talked about last time, which has just the chromosome label and the chromosome length, tells us when to stop making windows. Okay. So this is what a genome file would look like. We'll download this one that I set up for class. There's another one in there that we downloaded last time, but let's just do what this says. So it's human. I like this way of naming things better because it tells us what version of the genome and what species, because the chromosome lengths will change from version to version within a genome build. So, for instance, if we're focusing on chromosome 22, we will start at 0, and we will not have a window past 51,300,000 base pairs. Right? So how do we do this? There's a command called make windows. And all we do is tell it the window size, 10,000 base pairs, and the step size. So we're going to step 5,000 base pairs along the way. We give it that genome file, and we hopefully get good things. Oh yeah, okay, fair. Right? Anyone see a problem? There is a problem. It's chromosome 1, but I'm just looking at head. Chromosome 22 flashed by. It's a subtle problem, and it's, whoops. So these are 10,000 base pair windows for chromosome 22, but we now have this same CHR 22 versus 22 problem. If we go to intersect this file with VCF file, hilarity will ensue. So let's fix that and let's use our find replace trick. And let's use our grep trick. Let's use awk. Okay, and let's make a file out of this. Looks like I did that there. Oh. Oh, I see. So I named it as it's human, build 37, chromosome 22, 10,000 base pair window size, 5,000 base pair step size dot bed. Right? Okay. Same experiment now. We are literally just taking this different bed file. That's 10,000 base pair windows, not enhancers, intersecting with a VCF file, counting, computing a density, and landing a new file. Whether or not this works, we'll see, but let's roll with it. Seems like it did something. Trust but verify. Huh. Lots of zeros. Lots of zero counts. Lots of zero density. Like lots and lots, and then all of a sudden we have something. What's going on there? Anyone know why there's so many zeros? Yeah, so 22 is an acrocentric chromosome. So the first like huge, like many megabases is all ends. So there's no information. So we created windows for the acrocentric centromere, but no variance will ever be there because the sequence aligner cannot align FASTQ sequences in the FASTQ file to all ends in a reference genome. If you can't align data there, you can't find genetic variation. So this would be an artifact. Like if you printed that distribution, we'd see all these 10,000 base pair windows with zero density, and we might get excited. But then this is where visualization really helps. If we just took those bed files, this bed file with all the densities, and put it up in UCSC Genome Browser and look at it, you would see that all those windows with zero are in the centromere. Okay. Okay, so that is evidence of a false positive constraint estimate. It's zero. That is the calculation, but it's zero for artifactual reasons. Okay, so that's what it would look like. We get this big spike at zero. It's basically just doing the same thing in R. But then, everything that's not zero, that's probably where we can start. We'd want to almost exclude everything that's zero here because there's the acrocentric centromere, but I also told you in lecture 3 or 4 that there's still gaps in the human genome. Some of these 10,000 base pair windows overlap regions of the chromosome that are yet unassembled. We still don't know what the sequence of some of those are. So I would say for this analysis, this particular analysis, where we're breaking up the chromosome into chunks, we should be very, very suspicious of anything with a zero. It's a slightly different scenario for the enhancer thing because those enhancer sequence regions are probably never all ends or gaps in the reference genome, because Chrome-HMM predicted it was an enhancer based upon the alignment of some RNA-seq data or some ChIP-seq data. So it's almost certainly not a gap. So different consideration depending upon the data that we're studying. You with me? Okay. All right. So there could be things like different repeat content, things about the chromosome. Basically, this analysis is to show you that actually a lot of these regions are just ends because of a gap. The only thing I want to highlight here is a tool that I don't want to spend too much time on, but it's called getFastA. And what's cool about it is this bed file that we have for 10,000 base pair windows, you can give it a bed file and a FASTA file, the full reference genome. And for every interval that's in the bed file, denoted by this orange, it'll go in and yank out the nucleotide sequence associated with it. So for each interval, you actually know the nucleotide sequence. So if you're interested in like transcription factor binding sites or stuff like that, you could yank out all the different nucleotide sequences for your enhancers or these 10 kb windows, and then do some downstream analysis to look for particular motifs that you're interested in. The point of it for this exercise was really just to show that, yeah, if we go look at some of those interesting regions, guess what? They're ends. Okay. So this is more just detail about why we might be suspicious of these. There's another command in bedtools called nuke, which will allow you to profile the nucleotide content, how many A's, C's, G's, and T's there are for each of these regions that we've identified. And we can use this other command called bioawk, which I won't get into today, but it's basically awk developed by this Hung Lee again. But instead of referring to columns by number, so $1, $2, $3, if your input file actually has a header, you can refer to columns by name. So this nuke program from bedtools, it prints out columns. So these are the first 6 column, this a header with column names. These are the columns that were provided in the input, and then it adds its own columns, computing the percent of nucleotides that were A or T for that interval, G or C for that interval, the absolute number of A's, C's, G's, T's, N's, other nucleotides, and the length of the sequence. This header line, you can actually refer to these columns by name in bioawk if there is a header line. So it's basically a fancy version of awk. It's actually quite nice. And it not only has this behavior, but it'll work just like original awk. So if you wanted to, and I would actually recommend this, I have this set up on my own laptop, I just alias awk to be bioawk. And that way I can do sort of the normal refer by column number, or I can, if I have a header, I can refer by column name.
I have a question.
Yeah.
So would that method be better than just like figuring out like where the centromere is supposed to end and then just making a new?
Could do that too.
Chop it off?
Yep, absolutely. So you could definitely do that. If you knew where the interval started and ended, you could define a separate file which has one entry in it, centromere, chromosome 22, start and end, and then you could intersect your Windows file with that, but use the -v option. So they only retain the windows that are not in the centromere and then redo the analysis. Absolutely. So I want to move on to now. This is the thing I really want to focus on, and we might have to go into a little bit of the next class to really think through this. It gets at this question of if we account for this centromere issue, remove all the ends as was just mentioned, and we calculate variant density for each of these 10,000 base pair windows. I mean, ultimately, if you're going to convince yourself that this is interesting and worth publishing, like, hey, here are 10 kb windows in the human genome that are constrained, we really have to assess whether or not it's statistically significant. And in other words, that's saying, well, what is the chance that that observation we just made could happen randomly, by random chance? What's the likelihood that that's just a random process that we're measuring? And that's a hard problem for basically all of biological sciences. That requires good controls, replicates, and all sorts of complexity, like, making sure it's repeatable by other people in the lab, making sure it's repeatable by other labs. So I just want to do one experiment. It looks like we won't get through all this today, but that's fine. We'll pick up. Well, I guess not next week. You guys are gone next week, right? There's no class next week? Okay. So we'll pick this up later. But what I want to show you, I want to think through this process of whether or not something is really a signal, that it's significant, or it could be explained by noise. It's random chance. So what we're going to focus on is taking this file that I talked about last time, which is all the SNPs that have been predicted to be genome-wide associated with some trait, and ask if those SNPs overlap enhancers in human embryonic stem cells more often than you would expect by chance. And the motivation for this is, in human genetics, there's been this long-standing problem of when you do GWAS studies for a trait that you think is heritable, the vast majority of the SNPs that actually seem to be significant are actually not coding. And so the idea is like, well, if these are really correlated with the trait, and that's really what GWAS predicts, it doesn't talk about mechanism, it's just showing correlation between an allele, a variant, and a trait. What might the underlying mechanism be that explains that correlation? And so the hypothesis underlying this sort of partly the missing heritability and partly the fact that variants tend to not be coding, is that these are in regulatory regions. And so it's this very subtle polygenic effect where you slightly perturb the expression of a given transcript that's needed in a particular organ at a particular time in development, or in response to things as we age in the context of like adult onset diseases. Slightly perturbing those regulatory elements, for example, enhancers is what's going on. That's still really an open topic. But one of the things that the ENCODE paper showed, they claim, is that these GWAS SNPs are actually, they do coincide with regulatory elements more often than you'd expect by chance. Subtly so, but statistically significantly so. So we're going to basically try and redo that same experiment and see if we believe it. Okay. Last time we downloaded, we have enhancers, right? We've been messing with that all day. Last time we also downloaded a file called GWAS.bed, I think that's what it's called, and that's just all these SNPs from all the genome-wide association studies. We need to do this experiment, we need to calculate the number of observed overlaps between the two. And what's new, everything we've done so far today is just what do we observe? The new concept here is, well, what do we expect? And so usually what we're doing is comparing what we observed, signal, to what we expect, noise. I think all we will get to today is calculating the observed. I'll revisit this in a week and a half or so and reframe this question, but when we get back, what we'll talk about is this expectation, really thinking through that deeply. All right, so we found our enhancers, but now what we're going to do is we're not going to limit this to just strong enhancers or just chromosome 22. We're just going to take all comers, and we're going to make a full genome-wide set of weak and strong enhancers. Okay, so we got them on chromosome 1. Let's just take a look and make sure. That's darn slow. Means it's probably wrong. Yep, because there's no file mentioned. I once ran a command that I thought was right and sat there waiting for it to finish for about 3 hours, and then finally realized that it was just waiting for me to tell it what to work on. There was no file mentioned. Embarrassing. So I just looked at all the enhancers by chromosome, sorted by the count in descending order. And as you might expect, the most enhancer is on chromosome 1. It's the biggest chromosome. Chromosome 2 next. But then looks like chromosome 6 and 7 have more than you might expect by chance, because they're slightly smaller than 4 and 5. All right, so got that file. I've got about 1.5 minutes. I think I will stop there. This is our all enhancer file. What we'll start with in 1.5 weeks is comparing this to the GWAS SNPs to get our observation and talk about expectation. And we're going to introduce something called the Monte Carlo simulation to actually develop an empirical expectation to compare these observed values. Okay? Have a great break.